Human Physiology Laboratory Manual. So this is the lab manual. Um, yeah, I mean it's yeah, we don't have to. I mean, this is actually these guys. These are the guys who actually they work together as a husband and wife team. Um, Carl Ferdinand Corey and Gertie Teresa Radnitz earned medical degrees from the German University of Prague in 1920. Married later that year, and they are the ones who figured out the whole mechanism for blood glucose regulation and insulin and all that. And they ended up winning a Nobel Prize. Our efforts have been largely complementary, and without the other would not have gone so far. Part of the reason I put this on the front is just to emphasize that part of our lab will be kind of working together. Part of it we will have like kind of breakout rooms where you can kind of discuss what's going on, try to come up with hypotheses. Other parts of it are going to be on your own, like the actual analysis and things like that. Like your specific write ups will be your own work, but there will be also parts of the lab that I'm going to try to maintain that idea of you're working together. In fact, it was interesting. I went, you know, science conferences now are obviously also all online and kind of weird through Zoom. And one of the keynote speakers at one of these things I was just at recently, she's just mentioning how she really thinks that science is probably really like taking a big step backwards in this year, just because so much of what happens in labs even is not just people doing experiments. It's people kind of having conversations, kind of riffing off each other, kind of floating ideas off each other, random conversations in the hallways, people calling each other out. Are you sure? What do you think about that? Blah, blah, blah. So a lot of science. We're going to talk about specifics in terms of scientific method and ways people come with hypotheses and la la la. But a lot of science is also this kind of conversation and people building off of each other's work, right? Actually, I can stop this for 1 second. Humans only live a pretty limited amount of time, and doing real science takes a lot of time. I spent like 5 years of my life in grad school, just in my lab work, trying to look at, I could say, like in the big picture, like studying how the ear processes sound. But it was actually, the bullfrog inner ear, and actually the bullfrog inner ear sacculus, which is actually doing low frequency sound, more akin to like kind of felt vibration, and looking at a specific aspect of that, even like how does the bullfrog sacculus, preserve frequency and timing information. And that was like 5 years of my life. Um, so most science, if you really want to get at the answer in a more rigorous way, you have to really focus in on a very small part. So you end up having lots of people working on very similar things and all kind of riffing off each other and adding to a bigger picture. And also kind of holding each other accountable. When we get into the idea of the scientific literature, people are constantly checking each other to make sure that they aren't forgetting something, missing something, not realizing their biases are influencing their interpretations. So there is a lot of that in science as well, where people are trying to hold each other accountable. So at some level, we're getting at some deeper truth about the universe, even though we're all just humans, which are flawed and biased mammals running around the planet. Um, so We're going to talk about these different processes involved in trying to, it's like you got, la la la, this is your book. This stupid thing makes it hard. There it is. Ha! It recognizes a human, and this is on. It says, like my t-shirt. This is your book. You're going to read all this stuff in your book about how the body regulates blood pressure or how neurons communicate with each other and all that. It's like, how do we know? It's not somebody took it and pulled it out their butt and it's like, oh, now we got the story and we understand it all. It was like, at some point, we have no idea. And people start doing, this scientific method and start building this kind of narrative about what is going on. But it really is useful to understand that process, especially as you're getting further on in your careers. You don't want to just be a passive person. Well, you just read it in a book. That's how you find out about the world. That's not. And this is actually not the last word. It's particularly physiology. Physiology is a work in progress every week. And the idea or the understanding of the details of your immune system keep changing. Every week, details about the details of your nervous system keep changing. The big pictures usually stay relatively constant, but the subtle stuff changes regularly, and sometimes even the big stuff changes. As you go out there in the kind of medical world and you're taught about what are the best practices. Here's somebody presenting with this condition and how should I approach it. What is the best practice today might be different than what we believe a year from now, because people are going to do more research, do more kind of studies trying to figure out what is going on, what is the best way to approach this, or what is the best way, diet, what is the relationship between your diet and your health. That's constantly changing and in flux, and there's lots of often contradictory information. So it really is useful to have a deeper understanding of how we figure out, explore the world, get information about the world. What are the strengths? What are its weaknesses? So that's going to be a big part of our lab, is learning that process of even initially formulating questions so you can get to an answer, designing experiments so you can get data that might hopefully give you an answer, analyzing that data, which is always going to be kind of unclear because the world's full of randomness. I'll talk about that in a little bit. But you rarely ever get definitive results. We're going to talk about this in more detail later. Like if you want truth with a capital T, you're never going to get that in science. Science gives you, this is our best understanding given the empirical data that we have and our tools for analyzing. So it's very powerful. It gives us a lot of useful information. It gives us tools that we can use to heal people who are sick or land people on the moon. But it still has, it has kind of built-in limitations as well, which are worth paying attention to. All right, back to lab manual. So this is the lab manual. Let me change my view here. Oh, that's not what I wanted to do. There we go. All right, so this is the beginning here. Lab procedures and expectations. So I might as well start here. So we can zoom in a little more. So a place to both explore lecture concepts firsthand and also a place to learn more about the process and discovery of science. So that's going to be important. There's two main kinds of labs that we're going to be doing. One kind of lab is going to be more of a demonstration lab. Like the lab we're doing on Thursday is going to look at different concepts in chemistry, but it's more about kind of observing stuff that we're learning about. And we kind of know what we should be finding because we know the basic theory behind things and what should dissolve in what and why. So some of our labs are going to be more about demonstrations of concepts to just make them a little more visceral. Other labs are going to be more about the process of discovery. Like we have a question and we want an answer. So how do we go about that? How do we design the experiment? Here's where we get the data. Here's how we're trying to analyze the data. So that will be two kind of different kinds of things going on in our lab. Some, again, that are more about just demonstrating concepts. Some that's more actually what we call like hypothesis driven, where you've got like a question, you have a hypothesis, and now we're going to test the hypothesis and get the data and then kind of analyze whether or not the data supports our hypothesis or not. You are going to have to keep a lab notebook. An important characteristic of a lab notebook is that it have all your observations, data, notes, comments in it. Should be a complete, concise narrative of your work, clearly readable, understandable. This is not going to mean as much because we're not meeting in person. In general, you don't want to write data on scratch paper. It needs to just go into your lab notebook so you know where it is, what the conditions were when you were gathering it. The very first page of the lab notebook is going to be a table of contents so you can find stuff. The lab notebook, in fact, it's going to just be easier if I get rid of my little magical bridge here. Hold on a sec. The virtual backgrounds, I like them, but they make it weird when you're just trying to show things. So, lab book. Everybody needs one of these. Again, these are also pretty cheap. These are like 10 or 15 bucks at most. It has to be this kind. It has to have binding. It can't be loose pages. The pages are numbered, 19, 20, 21. This way we know that you're doing things in order, and if things are out of order, we can see that. It needs to be this graph paper because you are going to be making tables and charts and stuff like that. So this is a very specific kind of lab notebook that you need and that you will be maintaining through the semester. Again, it's got bound, numbered. It does not have carbon. There's some lab notebooks have like carbon copies and things. This shouldn't be like that. You're going to write on every page, front and back. The front page is going to be your table of contents. And you might ask yourself, well, if I'm doing this at home, how am I going to actually turn it in? And the way you're going to do that is using these little Adobe scan apps, or there's other apps as well, but you get them on your phone. You basically take pictures of the pages and it kind of easily assembles it into a PDF file, an Acrobat portable document file, and then that's what you will submit on the Canvas as your lab. I'll show you examples. I just downloaded some from last semester so you could see this. Where did I just put it? Here it is. So I'm going to share again here. So for instance, table of contents. What? Hold on, I hope this thing didn't spaz here. Here we go. So this is a table of contents. This is showing, and this is something that's going to be a work in progress, right? You don't know. In fact, we don't know. We tend to shift the schedule as time goes on, depending on how things are going. So you do know. In fact, actually, I didn't actually tell you about this. Thursday is the jumping lab. How do we know it's the jumping lab? Because there. Because I got it. Where do I get it? It's up here. There's this schedule. This is on the syllabus homepage as well. And here it says for each day, what is the topic we're going to be covering? In general, it changes. And what chapters from the Silverthorne book are addressing those topics. And then what lab we are doing that day. So Tuesday, January 26th, it's introduction. That's what we're doing right now. Introduction. Thursday, next class, we're doing chemistry review lab. So, and this is my tentative schedule. Like I said, it might change a bit, but this is in general what it's going to look like. Labs that don't have any kind of highlighting, those are the demonstration labs. Those are the labs where you don't really have to do much beyond. You should read ahead to know what the lab is about, but you don't have to prepare anything formally for those labs. Like for the chemistry lab on Thursday, you don't have to have anything done ahead of time. You should, aside from just kind of looking it over so you know what to expect. Some of these other labs, these labs that I've highlighted in this magenta, these are the labs that are more hypothesis driven, where you actually are going to have to come up with an objective and a hypothesis. I'll talk much more about that. As you can see, the first one isn't going to be until next week, so don't stress about that yet. I'll talk much more about the expectations of what you need to do for these labs that are highlighted in magenta here. And just for completeness, I should show you mid-unit quizzes. I have them here in aqua. These are like these little 10-point quizzes that are just kind of checking in on you. The green things, exam 1, 2, 3, and exam 4, those are those major 100-point exams. And they are going to take the entire class. You can see there's no other topic for lecture. There'll still be lab stuff on those days, but no new like lecture material. Exam 4 is happening during finals week. However, it is just another unit exam. It's going to be covering reproductive, endocrine, digestive, body defenses, and actually urinary system. But it's not going to be in depth in the other stuff. Just like each of these exams, is going to include stuff that's foundational. Like you still need to understand basic chemistry to understand the endocrine system. But exam 4, exam 3, exam 2, exam 1, they're not what we'd call like comprehensive in that they probe in depth every concept in the entire semester. Like I said, there will be the optional comprehensive exam if you need that to try to bring your grade up. If one of these initial exam 1, 2, or 3 ends up being lower than you would like. But these are the exams. I try to put, I put this after spring break because that gives you time. Hopefully it's not there to ruin your spring break. It's to help you be more relaxed. Anyway, so the labs. But back to Where did I? Oh, the PDF. The table of contents here. Hold on, I have a battery issue. Let me do this real quick. Blah, blah, blah. Each experiment will have a name and a page and a date. And again, for Thursday, it's only going to be jumping lab, probably on page. Not it's going to be chemistry review lab. I'm sorry, it's going to be your chemistry review lab. It's probably going to be on page 3 because page 1 is going to be your table of contents. And as we go through the semester, this is going to become more and more filled out. Right. This is what his table of contents looked like halfway through the semester. Your lab notebook is going to be useful for a few reasons. One is, it's a big part of your grade. Like I said, it's about 20% of your grade. But it's also something that you're going to be able to use during the lab exams. The lab practicals are open notebook. You can't use random notes. You can't use crib notes. But you can use your lab notebook that has your data, your hypotheses, your analyses. So doing a good job on your lab notebook is going to be this gift to your future self, right? If you do a good job on your lab now, you're doing this chemistry review lab and writing what everything means. And 2 months from now, you're taking the lab exam and it's asking you some detailed question about dissolving some such and some other stuff and what's going on and why. You can go back. It's like, oh yeah, chemistry review lab. Let's go look at this. What was I doing there? Read my analysis. What's going on? So this is going to be this great gift to yourself in the future as well, to do a good job on this because you'll use it during your lab exam. So this is the exception to like no notes on exams. For your lab exam, you actually are allowed to use your lab notebook. Your lab notebook, not somebody else's lab notebook, not lab notes, not crib notes, but this core thing. And just to give you a sense of what the labs end up looking like, when you actually do labs, you're going to be writing things in here. And then you do them in this, using this little thing in your iPhone to take pictures, and it assembles them as PDFs, which you then turn in. This one, the jumping lab, is going to require objectives and hypotheses, sources of variability. All labs are always going to have methods. The methods you can just cut and paste from your lab manual. There's going to be graphs, things that we're going to be using a lot of Excel. You're going to print out graphs. You can just paste those in, and then you have to make sure that you always number your graphs and then refer to the graphs in your discussion. So it's not just random pictures stuck in here. They have to be numbered and labeled well and then integrated into your discussion. But this is kind of what your labs are going to look like when you turn them in. This is how I tend to see them. Instead of actually having the physical lab in front of me, I will be getting these PDFs. That's kind of the mechanics of how we kind of dealing with. You still need to have a hard copy, but you're going to turn it in this way. Let's see what else I want to say here.
Did you mention how many pages it's probably going to have by the end?
Yeah, it'll probably be filled.
But so what kind of size? Because I'm looking at them, they have different page counts. How
Uh,
many?
this one here has 152. This is the classic one. Yeah, 152. It's like probably, yeah, double-sided though, so it's like it's probably 75 pages, but 152 sides.
I also have a question.
Uh-huh.
Um, on when you go on the Com bookstore and it lets you enter the course, so for example, your course, it says there's 3 things required. Do you want all 3 of those things?
What are the 3 things?
There's well, one says the lab book, which is what we're talking about right now, which I already ordered. And then one says Bio 224 Human Physiology for $14. I don't know exactly what it is. It has no image
So
and you can't.
that is the lab manual that we're talking about. That's what.
Okay, and then there's also a Human Physiology 9th edition by Sherwood.
Oh, that shouldn't be. So remember, we're getting the book for free.
Okay, I just wanted to make sure. I didn't get it. I got the other 2 already, but I just wanted to make sure that wasn't, because it says it's required, so I was a bit confused.
Oh, so I should go and tell them to get that out of there. Yeah, Sherwood, Laura Lee Sherwood wrote the book that I was using a few semesters ago. I like her stuff, but the Silver Thorn is also good, and you guys can use it for free. So I was like it was kind of a no-brainer there. Um,
Okay, thank you.
and all the reality is, any of the good college level human physiology books, they're all kind of different, but none of them are actually better. They're just kind of different. So yeah, so thanks. Yeah, thanks for letting me know that. So yeah, don't get the Sherwood. The Sherwood was the required text before we went to this loaner textbook program. Let me put a note to myself to get that out of there. Okay. So then there's this idea of what the lab write-ups need to be. It's a guy object. So I'm going to actually go. What time is it? We're going good. I'm going to go back to my little whiteboard here. And let's talk about, so for the labs that are hypothesis driven, these are the ones that I mentioned that are in magenta in your in the schedule. Right, so this does not include the chemistry review lab that we're doing Thursday, but will include the jumping lab that we're doing the next week. So for these are going to require a pre-lab. And I'm going to call it pre-lab kind of in quotes because it's actually just the first part of the lab, but I just want to make sure that you do it before you actually meet for the lab. And again, we'll be discussing the lab before this has to be done. I will be talking about the lab ahead of time, so you aren't going to be doing this out of the blue. I will orient you so you know what you're doing. So the pre-lab is going to require an objective to start with. And let's talk about this a bit. Even though this word sounds like, it can be just a regular old word in conversation like, my objective here is to do well in this class and then get into a nursing program and enter the field as a skilled healthcare provider, whatever. In this class, objective is going to be much more defined. This is the question that we are trying to answer by doing this experiment. So the question we are trying to answer. The question is almost always going to be of the form, How is some independent variable related to some dependent variable? Or, so which means that we need to start dealing with independent and dependent variables. So, because at the core of understanding how to think about these experimental designs, you got to think about independent and dependent variables. So let's take a step back and talk about independent and dependent variables. Dependent. So it's useful just to kind of think about like an example. Let's say I am interested in how drinking certain amounts of coffee affects people's performance on their exams. So I want to look for some relationship. Like how does the amount of coffee you're drinking affects how well you do on your physiology exam? And maybe I get a bunch of data. Like, this is like number of cups of coffee per day. This was score on exam. I should mention while we're here even, I'll mention this many times before we're done. Always label your axes. So then cups of coffee, let's say, 0, 1, 2, 3, 4, 5, 6. And then I get a bunch of data. I look at, there's Lily. Lily like drinks like 4 cups of coffee per day on average, and she nailed the exam, and so there's a 4 cups at 100. And then there's Paige, and Paige drinks like a couple of cups of coffee, and maybe she got a 90 on the exam. So there's that. And it's like everybody, we pick different people, and everybody's going to end up on this graph. Right, you've seen this in Bio 110. Parts of what you need to know from Bio 110. What do we call this kind of a graph? If you take lots of dots and put them on there based on these two axes here. What kind of plot is this?
Is it a scatter plot?
Yeah, exactly. This is a scatter plot. And then I can look to see, is there some relationship? If I look here, I can say it's kind of messy, but in general, it does seem like the more people have been, let me erase a couple of these to make it even more obvious. The more coffee people drink, seems the better they tend to do on the exam. So in this case, which is the dependent and which is the independent variable?
The dependent would be it would be the cups of coffee. Sorry. Is that correct?
Score on the exam?
Yeah, so in general, the independent variable is a thing that is being varied. So you are trying on different values of this. you check different values. And then the dependent is look to see if it moves in some predictable way. As the independent variable is being varied. So in this case, cups of coffee, we're looking at different values for how many average cups of coffee. And then we're looking at the dependent variable, the score on the exam. Does the score on the exam seem to vary in some predictable way as the average cups of coffee drank changes? Does that make sense?
Yes.
And in general, the dependent variable is always going to be on the y-axis, the abscissa, and the independent variable is typically, no, I'm spazzing. This is the ordinate. The abscissa is the x value. So the x value is going to be the independent variable, and then your dependent variable is typically going to be the y-axis. So that's kind of the classic way to do this. So this is what would I call this relationship here? If in general, as the independent variable goes up, the dependent variable also goes up.
Positive correlation.
Correlation.
Yeah, so this would be a positive or a direct correlation. Right? It's also possible that it looks like this. What would I call that?
An indirect relationship?
This would be an inverse or negative correlation. Right? Because the more, and this could also happen, another thing that's really common, even though it's disappointing, is this. Something like that. What would we call this?
No correlation.
Yeah, this is no correlation. Uncorrelated. Right? I mean, this is where being a scientist, you have to be very careful with your biases, because usually people have hypotheses, they believe in it, they're pretty sure they're going to find what they want to find. And sometimes what you find is like, actually, you're wrong. It's not there. And yet, we'll talk about this as we get into the nervous system. Your nervous system is really good at pulling information out of the noise. And sometimes it even does it when there is no information. It makes stuff up. So you have to be, especially if you're expecting something. So we're going to talk about basic statistical tools and stuff that try to analyze whether or not there really is a correlation or not. And so these are kind of linear. For our class in general, we'll stick to like linear relationships, kind of positive correlations, negative correlations, uncorrelated. In reality, most things are more complicated than that. It's actually, even for this example here, it's very likely, that it's going to go up. But then at some point, if people are drinking so much coffee that they can't even hold their pencil straight, it's going to start going down. So it might actually be a non-linear relationship. Might be more of some second or third orders more to look. It might not be necessarily just purely positive or negative. Just kind of putting that out there. In reality, these relationships aren't always so obviously like predictable. Other things we can talk about while we're here. Even if there is a positive correlation, let's say we do the experiment and we see like, sure enough, as people drink more coffee, they did better on the exam. It's always important, again, when we talk about the strengths and weaknesses of the scientific method, this does not prove, we'll talk about for a number of reasons, it doesn't prove that drinking coffee helps with your exam. You know, we're never going to use the word prove. I'll talk about why. So one thing is correlation does not imply causality. You know, yes, people who drank more cups of coffee did better on the exam, but was it really about the coffee? You know, it might very well be that actually it was people who studied more in groups and supported each other did better. But, you know, the people who in the study groups were meeting in cafes, and they were in the cafes, let's say this was pre-COVID, and then they're like feeling guilty that they're sitting there for hours on end and not supporting the business, so they're buying coffee. But the coffee had nothing to do with whether or not they did well on the exam. The coffee happened to be incidental to the fact that their study groups were in a cafe. So sure, yes, more coffee, people who drank more cups of coffee on average did better on the exam, but it really wasn't about the coffee. It was about their study groups. That's possible. There was one example of a study looking at whether night lights made it more likely that you're going to wear glasses as you grew up. And they found that homes that had more lights on at night, that the kids were more likely to have glasses, as you know, need glasses eventually. And so they're thinking, oh, maybe there's something about having, you know, when we were like, you know, just, you know, animals in the forest, you don't have all these artificial lights everywhere. So maybe having lights on at night's messing with our development of our vision. And they found like, okay, more lights, more likely to wear glasses. But then they found it actually had nothing to do with the lights. They found that it was genetic. That people, that whether you needed glasses was most related to genetics, whether or not your parents wore glasses. And if your parents had worse vision, they were more likely to have more lights on around the house so they could see better. So there were indeed more night lights on at night for the kids who ended up wearing glasses, but it wasn't because the night lights made them wear glasses. It was because they got the genes from their parents who then were more likely to put on night lights. So just kind of putting it out there. Sometimes making an interpretation of what does the data actually tell you, what does it mean, you got to be careful. You don't want to just get on your soapbox and say, I just found this and this is the answer. You know, sometimes it can be kind of tricky as you start looking deeper and deeper. So other things we can talk about while we're here. This randomness, if you take a look, you know, obviously, even in this graph I'm drawing here, even if it looks like, yes, on average, the group that has more coffee is doing better on the exam, you can see that it's messy, that the actual data is scattered around. There's a lot of randomness in the universe. If you, even you, like next class, we're going to do this jumping lab next week. If you do the same jump exactly the same way 3 times in a row, you're not going to jump the exact same height, right? You're going to jump a different height. Like if you try to do anything, there's nothing, things don't always work out exactly the same. So So this is a scatter plot. Let me show you a different plot. Let's say, let me do a slightly different version. Instead of doing this more continuous thing of average cups of coffee versus test score, let's just say coffee drinkers versus non-coffee drinkers. So now instead of doing these continuous variables, my independent variable is now a category. Right? And I could do some graph here. And if I looked at kind of average exam score, maybe it looks like this. What do I call this kind of graph where you actually are comparing categories of things rather than looking at the scatter plot between two continuous variables?
Bar graph.
The bar graph. So a bar graph, you're using that whenever you're comparing categories of things rather than things that are quantitative. This idea, this average, this is going to be important. Average, this is what we call a measure of central tendency. Actually, let me make this less messy because this is getting messy. Where's my little... Where has my... Huh. I'm not sure where my little keyboard... Oh, there's my little keyboard. All right. So this is what we call a measure of central tendency. So in reality, if we look at all the coffee people, some of them maybe got A+ with extra credit. Some of them maybe didn't do well. But average, how do you, and the same thing for the non-coffee drinkers. There's different values, but you have some average. How do you calculate the average of a bunch of data like this?
Take the total score and divide it by the number of people who had scores.
Wait. So yeah, basically, yeah, you would add them all up and then divide by the number of subjects, right? So average, this is one of the main ways. There are different ways to get measures of central tendency, but we're going to be using the mean average in our class. Right, there's different other averages. Those of you who have been in in the housing market know about the median home price. Median is another kind of average. It's like what value or half the house is more expensive and half the house is less expensive than. There's different kinds of averages. We're going to be doing mean average, which is just basically add them all up and divide by the how many subjects. So measure of central tendency allows you to say, in general, coffee drinkers versus non-coffee drinkers. It looks like the coffee drinkers did a little better on average than the non-coffee drinkers. So measures of central tendency let you kind of get a value for something that actually is kind of spread out and random. But now the other thing that we want to do, if we actually want to be able to compare, like, okay, yes, this is a little taller than that, but does it mean anything? Is it actually meaningful? So the other thing we need to do is measures of dispersion around the mean. So let me do two examples. Hold on, I gotta make this. So in both cases here, we have the coffee people have a slightly higher average. This is supposed to be average test score. In both cases, the average test score for our coffee drinkers is a little higher than the non-coffee drinkers. But to actually responsibly interpret that, you can't just go and say, well, sure looks like coffee helps, with your test scores. Because you also have to take in these measures of dispersion about the mean. So how spread out is the actual data? Like if I look at my actual data points, it could be they look like this. Here's the mean, and the range of the data around it is really very tight around that mean. In which case, I'm more likely to believe there is an actual difference between these two groups, and that the difference between those two averages is telling me something interesting. It could also be that the range looks like this. Yes, the mean on this group is a little higher than the mean on this group, but if I look at, this group had lots of people who failed and this group had lots of people that got 100%. It's just when you do the averages, it just so happens that the average of these numbers ends up being a little less than the average of these numbers. So this, we have the range, the data is very tightly grouped around my average. Here the data is really spread out around the average. Which one of these is telling you something useful, probably? Is this giving us any information about the relationship between coffee versus non-coffee and how well you'll do on an exam?
No, not really.
It doesn't really, because there's so much wide randomness in both groups. We don't really trust that this average really means anything. So we're not going to, if you've taken statistics, you've probably done like the student t-test. You can actually there is a statistic you can calculate where you tell it what are the two means. What is this, measure? One of the measures of dispersion is standard deviation. That's a classic way to do measures of how spread out is the data around, 95% of the data points are within 2 standard deviations of the mean, right? It's a way of how spread out is the data. But you can calculate these statistics where you tell it what is the dispersion, what's the standard deviation, how spread out are these, and it will tell you how likely is it that this difference is real versus not real. It might say that there's like a 50% chance that the difference between these two averages is just due to the randomness in populations that you'd expect. Even if we just took people who were exactly, everybody drank coffee and we took half of them and took the averages of the two halves, it still could be spread out that different. Does this make sense?
Yes.
So In our class, you're not going to have to actually calculate, the more formal statistics like standard deviations and t-tests and things like that. But you will have to do kind of more of just a ballpark. One way we can do measures of dispersion is just the range of the data. Right? If I look at the range, the highest data point to the lowest data point that I used to get that average, and I see that they're both spread out pretty much the same, that is going to be a clue that this difference between these two means is probably not really meaningful. Whereas if I calculate, if I look at the ranges and the ranges are really tight and the difference between the means is kind of far, then I can say this probably is telling me something about an actual difference between coffee drinkers and non-coffee drinkers. So you can make errors. Because doing all these calculations, all these statistics are about probabilities. So the statistical calculations that we're not going to be doing more formally. We might actually. It depends how much time we get. But they tell you how likely is it? What is the probability that the effect you're looking at is real and not just due to the randomness that you're always going to find? So the two kinds of errors you can get are sometimes you look at this and you're like saying, the data is so spread out, we're going to say there is no effect here. We can't responsibly say that coffee drinkers and non-coffee drinkers are different based on the data that we got here. But maybe there really is an effect, but it just got lost in the randomness. Maybe if you took more data points and did a more, kind of tighter kind of analysis or whatever, you'd find out eventually that there is something real. But one of the errors you can make is saying there is no effect when in fact there actually was something. And obviously the other error is going to be the opposite, where you say, well, I'm doing the math and it's like, these are pretty far apart and the data is really clustered around the averages, and you do the math and it says, yep, it sure looks like coffee drinkers are doing better on average than non-coffee drinkers, but in fact you got fooled by the particular subjects you got made it look like that, but in reality, if you got more and more subjects, that effect would go away. So you get these, they're called type 1 and type 2 errors, where either you see an effect when there's actually no effect, or else you miss an effect when it actually is there. So this is part of the reason I'm doing this, is kind of showing you some of the trickiness in science. Science isn't just doing an experiment, getting your data and knowing the answer. The next step of how do I interpret this data is really tricky and leads you into trouble too. Up till very recently, this was the kind of gold standard for being able to write up in a scientific journal or paper that your effect is real, that what you're seeing is real and not just random. You do the statistical calculations and the statistics tell you, I can say 95% sure, with 95% confidence that, this is difference in test scores is due to the coffee and not due to randomness. And then you get to publish that, and it's called statistically significant. If you can show through the statistical calculations that we have a 95, greater than 95% chance that this is real and not just due to randomness. Obviously, what's the danger there?
There's still a 5% chance you're wrong.
Exactly. You're putting this out there and saying that this is very likely true, but that means there's still a chance it's not true. Again, this is where science is very different from like religion, where it's absolutely true or not true or this or that. Science is always talking about probabilities. It's likely. How likely is it to be true? And it also means that sometimes, that's why the stories change as we get more data, more information. And also, I'll talk about this in more detail later when we talk about scientific method in more detail. This has actually led to some real issues around people just getting something and then wanting to move on because they want to continue on and publish more papers. But they've actually found like some disturbing things now. More recently, there's been people going back and recreating old studies to replicate their results and finding that they don't replicate nearly as nicely as you wish they would or you think they should, because just because they got a good result this time, if you do it again, you might actually find out that it's not consistent. So you have to think about it. Alrighty. So 11. I've been yapping a bunch here. Maybe we'll take another little break, and I'll continue talking about the about independent and dependent variables, and then talk a little more about the expectations for the lab notebook. But we're going to be seeing these ideas again and again. But that's part of what our lab is about, is not just doing stuff, but actually understanding the process that people use to, how do we know everything in this book? How do all the things you're going to be learning in your nursing classes, how do they learn that? How do they know what the best drug or the best this or that is to do in a certain this or that situation? It's all based on these processes. And the more you understand that, the more you can responsibly decide whether or not you trust what they're telling you or you don't. And you'll also be able to find your own answers. There'll be something you're curious about and you want to figure out what's the best way to approach something. Having these tools will make it a lot more likely that you're going to kind of arrive at the best possible kind of solution and outcome based on kind of the information that you've got. So it's 11:15. Let's come back at 11:30 and we will continue with all of this. So continuing on our discussion of its objectives, because this was all kind of riffing off of this idea on the labs where we have a hypothesis-driven thing going on. The objective always going to be a question, which means it should have a question mark after it. And it's always going to be something about relationship between the independent and dependent variable. So that's why it's going to be really important that you kind of get clear on what is the difference between an independent and dependent variable. Like, for instance, the jumping lab that we are going to be doing in class next week. The basic idea of the jumping lab is you measure different body measurements. You're going to measure people's height. You're going to measure the calf circumference. You're going to measure their weight. And then you're going to have them jump up against the wall and see how high they can jump. And we want to see if any of these body measurements are predictive of how high that they can jump. So that's the basic idea. We're trying to see how the vertical jumping ability of somebody is related to how much they weigh or how tall they are or their calf circumference. So what would be the independent variable and the dependent variable in the case of this jumping lab?
The independent variables would be the height, weight, and calf circumference, while the dependent variable would be the jumping height.
Exactly. So, and notice she also made a kind of got to this more confusing thing. There's 3 independent variables in this one. There's the person's weight, there's the person's height, and the person's calf circumference. So you're checking out different versions of weight and see how it affects jumping height, different heights, and different how does it affect jumping height, different calf circumferences, does that affect jumping height? So in this case, there are actually 3 independent variables and a single dependent variable that you're looking at in each case.
So then would you find the mean number of each variable? If you like, let's say there's 10 people.
This would be more likely to use a scatter plot, kind of like we did with the coffee, right?
Okay.
You could do like, this is their weight, in kilograms. This was their jump in centimeters. And then for each weight and each, you put what was the jump height for that person, their weight and their jump, and then seeing is there a correlation or not between height,
Okay.
between weight and their jump height. We're also going to be analyzing the data for BMI, body mass index, which is a formula that is related to their height and the weight, in which case then you would have a bar graph, right? If I'm going to do like healthy BMI versus unhealthy BMI, and average, then I do average jump, and then I could do it like that. That would be a bar graph. Or if I was going to do male versus female, it's another group that we can use when we're comparing our data for this jumping lab. In that case, you are getting these averages. And then you do want to kind of see how spread out is the data around those averages to see how much you trust them. Chris was mentioning a few moments, he messaged me about whether or not you're expected to do standard deviations in this class. And the answer is no. I'm not assuming that you are comfortable or familiar with kind of more formal statistical calculations beyond calculating a mean average. But looking at just the range of the data there's no math involved. It's just what's the highest and lowest data point that you use to derive your average from. And it can give you a sense of how spread out the data is. If you were going to do something more formally, you would have to start using the statistical calculations. But just looking at the range of the data gives you information about how much you trust your results. Telling you why this is important. Let me just give you a quick little example. We're not going to actually do independent study groups or independent study, given that we're in COVID and Zoom land. We have a different research project, but it's normally we have students do their own experiments around things that they're interested in, and maybe there'd be some graph like this, like change in blood pressure, from resting, with yoga group versus the non-yoga group or whatever. And they'll do something like this, in millimeters of mercury, and they'll say like, oh, it looks like this yoga group had like less of a change in blood pressure when blah 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 blah, and they'll draw it like this, and people will look at it and say, oh cool, it looks like something's happening, when in reality it's totally misleading. Like if you look at the range of the data, like this is sure the average here, but if you look at the big picture, it actually looks like this, with the range of the data like this. And if you just take this little piece and you magnify it crazy, it's like, oh, look, there's a big difference. But in reality, when you look at it in the context and really comparing to the range of the data that you are using to get those averages from, you realize that this is very misleading to say that this is true, or this is actually significant, or this means anything. So it really is important to not just look at the averages, but to look at how spread out is the data that you got those averages from. But that being said, for our class, you do not need to do the more formal kind of standard deviation or standard error. There's a lot. We're not going to go into that. If you were going to publish something in reality, though, you would have to do that. And I would even say the statistics get complicated. Like when I go about this stuff, I even usually go talk to somebody who is more of an expert to make sure I'm not spazzing. And because most of these statistical calculations assume you're making particular assumptions about the distribution of the data or this or that. So doing this responsibly can get tricky. So blah 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 blah. All right, so back. I'm gonna now share. Boop boop be doop. Share screen. So we're back here. We can see objective. Again, I wrote this here very as much as clear as I could. It will almost be something like, will almost always be like, is there a relationship or correlation between the independent variable and the dependent variable, question mark. Or what is the relationship between the independent variable and the dependent variable? Or how is the dependent variable affected by the independent variable? Or something similar. Right? I can't tell you how many times we're going to probably find, like, as people write their first objectives, people are going to write things that aren't even questions, let alone a question that brings in the independent and dependent variable. It just has to be something very simple. Once you figured out what the independent variable is and the dependent variable is, you just plug it into this. What is the relationship between the independent and the dependent variable? Done. Stop. Zip it. Because you just want a nice, clearly stated question. This part is really tricky. This is the beginning of everything. Because if you don't have a clear question, then you don't know how to go about answering what you're trying to find. And it has to be very kind of specific. I could say, like, what's the relationship of diet to health? It's like, that's way too general. I don't know where to go with that. Like, if my independent variable is like diet, what you're eating, I don't even know where to start there. So you need to really focus it in on having an independent and dependent variable that you can actually measure in some way, right? If I have a independent variable like, fiber intake in your diet, and my dependent variable chance of colon cancer, I can do that. What is the effect of fiber content in my diet and the chance of, getting colon cancer? That I can do. But unless you come up with a good objective that is focused and clearly stated, you're just going to be like spinning around trying to like do your experiment or design an experiment, because you need something more limited and focused in order to actually study it. So this objective, having it really clean, kind of concise is important. That's an important part of asking scientific inquiry is a very clearly stated question that you can then go about trying to answer. Hypothesis is just your what you think is going to happen. It's your educated guess. We're okay here. So educated guess. There's two things that are important in this definition. One, it's a guess. It might be wrong. You don't know what the answer is. If you knew what the answer was, there's no reason to do the experiment, right? If you know what the answer is, why do the experiment? But educated means you can't just be pulling this out of the air. You've got to actually have some reason. Some reasonable why. Some reasonable reason. It sounds weird saying a reasonable reason. But you can say, I think people who drink more coffee are going to do better on exams because coffee is a stimulant and they're going to be more awake. They'll be able to study longer with more focus and therefore do better on the exams. Or I think people who are taller are going to be able to jump higher because longer limbs will give you actually longer muscles that are probably stronger and longer moments of inertia to generate this upward force against gravity. So you who knows what's true or not? It's a guess, but there has to be some reason that makes sense. So educated guess. And the other part of these pre-labs is sources of variability. So there's always going to be the objective, which is basically the question that you're asking. Hypothesis, your educated guess. And then the sources of variability. And this one's important, particularly because your objective and your hypothesis are about this relationship between the independent and dependent variable, right? This is, how does independent variable affect the dependent variable? Sources of variability is what are other things besides the independent and dependent variable that might actually affect our results. And you want to think about this before you do the experiment, because sometimes you can actually control for them. If you don't think about this beforehand, your whole experiment might be useless. Like for instance, let's think about the jumping lab. The jumping lab where you are going to measure people's height and their weight and then watch them jump and measure to see if it's related. What are sources of variability there?
Shoes or no shoes?
Shoes make a huge difference, right? If you're wearing some Air Jordans versus wearing Birkenstocks or flip-flops, that's going to make a big difference in how high you can jump. Probably a lot more of a difference than your height or your weight even. So by thinking about that ahead of time, we make sure that all of the subjects are not wearing shoes. So we get that out of the way. What are other sources of variability with this jumping lab?
Maybe the surface you're jumping off of, like a carpet versus a hardwood floor.
Right? Now that could make a big difference. Actually, when I did gymnastics, we had these spring floors that really made a huge difference. So we're gonna have everybody, jump off the same floor. And speaking of that, just the mechanics, how you jump, how you move your arms makes a huge difference on how well you jump, probably way more than your weight or your height. So we're going to train people and make sure everybody's doing the jump with the exact same mechanics and form. What are other things that can affect someone's ability to jump that are not their height, their weight, or their calf?
Maybe like their energy level. So if it's like first thing in the morning compared to like middle of the day.
Right. So this is a good one because this is a variable that is probably making a big difference, but we can't control, right?
Okay.
But it's worth paying attention to that because you've got to think about it. And what our hope is that enough people are jumping a little higher than they might have because they're like really feeling like energetic, and it's going to be balanced out by the people who are jumping kind of lower than they might have because they're feeling kind of tired. So you hope that some of these sources of variability that you can't control, if they're random, then enough of the randomness is going to be above and enough is below that they kind of cancel each other out and just make things noisy, but don't actually shift the average value. But yeah, so some of the sources of variability, like shoe type, we can control for. Some things like energy level, we can't control for, and we just keep our fingers crossed that it's not causing a big change in things.
How about practice?
So practice for sure. Practice, and I was saying like the form of the jump. Other things, injuries. If somebody's like knees are blown out, that's going to make a big difference. So when we're designing the experiment, we would probably exclude any subjects who have any physical injuries that are going to affect their ability to jump. So when you are going to do an experiment, you really need to think about this before you do the experiment, before you get your data. Because otherwise, if you did not do that, then you get your data and people are going to say, why should I trust this at all? I don't know what kind of shoes they're wearing. I don't even know if they're injured, whatever. So if you can say, like, I did my best to control these sources of variability, and now the data that we do get is more likely to be what we're actually looking for. So this is going to be important.
When you say control the sources of variability, does that mean like your test subjects, you would want them all to wear tennis shoes, for example?
Right. Or in our case, we're going to have them not wear any shoes at all.
Right. Okay.
Exactly. Or we make sure all subjects are physically healthy and nobody has any injuries. Or we're going to make sure that everybody is jumping with the exact same form. We're going to describe where they have to swing their arms in parallel up as they extend their knees and things like that. So, all right. So what we want to do now, I'm going to share my other screen now. Okay, we're back to here. So I want go to modules. Whoop, wait a second. This is not what I meant to do. Hold on. This is what I want. Okay. So if you go into modules and you go all the way down to lab 0 orientation, there is something here that did not exist before, this dependent variables thing. And if you click on that, this is a study that actually came out this past year. It's actually kind of hilarious, kind of looking at supplements that you can take that might affect hangovers. So I figure this is a useful thing for college students. So this is what we call an abstract. We'll talk much more about this. An abstract is just kind of in a nutshell describing what the study was. It talks about what they were trying to do, how they did it, what they found, what their conclusions were. So what I am going to do is I am going to push you all into breakout rooms right now. There's going to be like 4 people per room. And in your breakout room, I want you to discuss amongst yourselves and come to a consensus. Everybody should agree with the same answer. What is the independent variable? What was or were the independent variables? What was or were the dependent variables? In a nice clear statement, state the objective of the study, and then tell me what were the results. What did they find? So again, this is more the kind of real world. Here's an actual study. I want you to be able to tease apart independent variable, dependent variable, and give me a nice, clearly stated objective. Are there questions about what you're supposed to be doing here? And you can reach out to me once you're in your breakout rooms. But what we're going to do is I'm going to send you away for the next 15 minutes, and I want you just to figure this out amongst yourselves.